Good evening, and welcome to Test 2, Cells and Cell Movement. Let's first start off with some cell movement. The word diffusion is pretty simple. It's the movement of materials from a high concentration to a low concentration without adding any extra energy. No ATP. In the picture here, you see there is a high concentration in the top left corner, and over time, these molecules will begin to spread. Now they can only spread until they've reached the edge of this particular container, but if all goes well, the diffusion will work until the distribution is even. Next, we're going to move on to the movement of material possibly through a membrane. And we have passive and facilitated diffusion. Once again, they're both in the diffusion family. So you will see that it is movement from high to low concentration with no energy. Now, passive transport is this kind of really big overriding theme. The passive transport is basically moving from high to low with no energy. Now, it can be what is known as simple diffusion. That is passive transport. It can be facilitated diffusion. That is passive transport, no energy needed. So if you look on the picture on the left, that is what is known as simple diffusion. And what simple simply means is it will come right through the membrane with no assistance, no protein channel, no extra stuff needed. This will be limited probably to extremely small, non-polar substances, things like oxygen, things like CO2. We're going to learn later that hydrophobic things like steroid hormones can also move through by what is known as simple diffusion, high to low. The other type of passive transport is called facilitated diffusion. Same thing, high to low through the cell membrane, but this time you need the help of a protein channel. Another example of this will be something called an aquaporin. This is how water moves across the cell membrane. Water, remember, is polar. It's small, but it's polar. So an aquaporin is another example of passive transport, but facilitated diffusion through what is known as an aquaporin. All of those things do not require any ATP. This next section does. Active transport is the movement of material against the concentration gradient, moving from low to high. In this case, you need something, and in the case of the cell membrane, you need a pump. A good example is the sodium-potassium pump. We're going to see a lot more of that in the nervous system, but the sodium-potassium pump is going to be moving sodium and potassium against their concentration gradient. But you're going to notice that you need ATP. ATP is going to break down into ADP plus a phosphate. That is where the energy comes from. It was in that bond. The ATP, as we know, will come from the mitochondria, but we didn't get there yet. Also important to note, the ATP and the pumping across the membrane against the concentration gradient, you will notice a conformational change in the protein. That is important. It just doesn't fly through. In the facilitated diffusion, the material simply moves through. It's almost like a tunnel. In this case, the pumping, there is a conformational change. In the world of active transport, active is not passive, and it is not facilitated diffusion. Both of those are in the passive transport section. Active transport is not the case. Moving to the cell membrane itself. The cell membrane itself is made up of a phospholipid bilayer. What that means is this thing is a phospholipid. There's the phospho end. There's your hydrophobic tails. That is one phospholipid. You can see there is a bilayer. embedded in that bilayer, there may be a protein. Those proteins could be active transport pumps. They could be facilitated diffusion. They could do a lot of things. They could be receptors. And what's going to happen is that regulates the in and the out movement through the cell membrane. We will see a lot more of that as we go. If we zoom in, you can see this picture. Well, it's kind of small, but there is that hydrophilic head and the two hydrophobic tails. This is the phosphate of the phospholipid. That's this area right here. This is the tail. When you put a bunch of them together, you end up with what is known as the phospholipid bilayer or the fluid mosaic model. And it's the fluid mosaic model because these are not locked. They can move. Water cannot stay here. These are hydrophobic. That's why you need something like an aquaporin to get the water molecules through. Also, you may see some cholesterol molecules in here. Now, the cholesterol molecules are going to help keep the plasma membrane fluid at low temperatures. Very important for animals that live where it is cold. Things like fish that live deep in the ocean, polar bears, whales, anything that lives where it's cold. Moving over here, we now see a U-tube. There may have been a typo on the original sheet, so make sure you change that to can't. In the first example, I have a setup, and it is important to always know the original or the initial setup. So in this case, my initial setup has 1.2 moles on the B side and 0.9 moles on the A side. So if the first part of the question, if neither the glucose or the NaCl can move, we're going to keep it that way. That makes side B hypertonic, which means water is going to flow from A to B, meaning the level of water on A's side will decrease, the level of water on B's side will increase. This membrane is important because this membrane is going to decide what can or cannot move through it. It will limit the size of the materials that can pass through. If we change that question just a little teeny weeny bit, here now I say that glucose can move and the NaCl cannot. Now that changes everything. Because of that now, instead of the glucose staying at 0.8 on B and 0.4 on A, we're going to split the difference. And you're going to see that the glucose is going to fall into its own equilibrium. That now messes up the overall concentration. Now, A's side is more hypertonic. So in this case, water will flow from B to A, causing A's side to rise or B's side to decrease. So it is very important to read the question. Does it start off as initial or after, let's say, 30 minutes or an hour? So very, very, very important to read the question. Sticking with that, basically what we talked about was the movement of water, which is known as osmosis. Now, osmosis is another version of passive transport. And it's another version of passive transport because it goes from high to low with no ATP. In this case, it's specific to water. That's what osmosis is. So if you take a look at these, this is your classic what would happen if to things like blood cells. If you put, and again, this is where the kids have a really, really, really hard time. If you put the cell, each of these cells, into a specific solution. So in the left picture, it's a hypotonic solution. That would be an example, let's say, of distilled water. Distilled water. And you know that hypotonic means it has less dissolved materials. So whenever you put any cell in a material that is hypotonic to that cell, water flows in. If it is an animal cell, it may burst. The exact opposite, let's go all the way to the right, if you put that cell into, let's call it a very high concentration of salt. very high salt water concentration. Now the outside is hypertonic, and what will happen is the cell will lose water to the hypertonic solution. I will say this many times. Water moves to the hypertonic area. Now the cell may be hypertonic, so then it moves in. If the solution is hypertonic, then it moves out. Read the question. If the in equals the out, and this is what you would love to happen in your body, that is called an isotonic solution. Plants have a nice ability to withstand extra water moving into them without them bursting, and that is because of the cell wall. Animal cells do not have that. This cell wall adds a lot of structure, it adds a lot of strength. When the water flows in and then begins to push out, it equals what tries to get in, therefore the cell does not burst, and that is called turgor pressure. And if a plant has turgor pressure, that means that its cells have enough water and life is good. Here's another example of what we did in class. In this case, mixed up the pictures. A, you know, is isotonic. A is very happy. You know it's happy because the water in equals the water out. Cell size does not change. In the second picture, in B, the solution, that's what is surrounding the cell, is hypotonic. That means it has less dissolved materials. Water is going to move into that cell. It could burst. That's why if you use an IV solution with distilled water, your cells could burst and you could die. In C, the solution is hypertonic. Let's say it is very salty. That's why you cannot drink ocean water. In this case, your cell is going to lose a ton of water, or the water will move out of the cell will shrivel, and potentially, again, you will die. This would be, let's say, distilled water or fresh water. This would be salt water. In this case, if there is a very large concentration difference, and here you can see it looks like there's a lot of solute. The larger the concentration difference, the more water will be either lost or gained depending on which way you're going. And we saw that in the potato experiment. So that is water movement, that is passive transport, that is active transport, that is facilitated diffusion. That is all anything to do with water movement. Coming up over here, a couple of more questions based on that. 5 cells, A, B, C, and D from the graph, were all placed in some solution, very similar to the potato experiment. The one that has no change in mass, you know that's the isotonic solution. So, for example, if I say the solution has a molarity of 0.3 moles, solution C must have 0.3 moles of concentration, and you know that because there was no change. If you look at A was placed in a hypotonic solution, and you know that because it gained mass. E was put in a hypertonic solution. You know that because it lost mass. A good example of that also is this picture down below. In the event that I put, like you did in living environment, I put some starch and some glucose into a beaker, into a solution. This squiggly thing, that's the starch right there. And the glucose is the little black triangles. Well, over time, I could use various indicators, and if I started out with a cell with no glucose in it, and my cell ends up with glucose in it, does that mean these pores were big enough to let in the glucose, but not big enough to let in the starch? Very, very important. The size of the pores or the size of the membrane, or if you're doing one of those fake cells, that is important. Water movement, done. Let's move our story over to cell organelles. Cell organelles, we learned a lot about those early in the chapter. Nucleus, as you know, contains the DNA. The DNA will then code for RNA. That RNA can leave through nuclear pores, move out to the ribosome, whether they're bound or they are free, and that was the whole endomembrane system, or just regular protein synthesis. Something like a prokaryote cannot do the leave through the nuclear pore thing because prokaryotes do not have a nucleus. They do, however, do protein synthesis. We will talk more about that later. Ribosomes, everybody's got those. Eukaryotes, prokaryotes, even some organelles inside of a cell. Things like chloroplasts and mitochondria. They have ribosomes. So if you have ribosomes, you can generate or create proteins. Again, if the ribosomes are free ribosomes, the proteins made will stay in the cytoplasm or in the cell, used for cell things. If they are bound ribosomes on the ER, or the rough ER, those proteins are bound to either end up in the cell membrane or exported from the cell in something like a hormone. Lysosomes get a lot of love here in this chapter. They are little mini, we'll call them bags of digestive enzyme. And what will happen is if anything comes into the cell that the cell does not want, can the lysosomes begin to digest it? Example, white blood cells that use phagocytosis to bring in things the body doesn't want. Lysosomes can destroy them. Also, lysosomes can be used to destroy entire cells in a process known as apoptosis, or programmed cell death. The rough endoplasmic reticulum does get a lot of love also. That is closely associated with the nucleus, and I'm going to try a really cheesy drawing of it, but you have seen that it looks like a folded membrane, and the rough ER, which is what we're talking about now, is studded with ribosomes. Those ribosomes are going to produce proteins, and again, the rough endoplasmic reticulum is going to produce proteins that are going to be exported or put into the cell membrane. Let's make a little change here. It is closely associated with the nucleus, but we're going to kind of change this a little bit. So we're going to change this to the production of proteins, which we just said. And those proteins are going to ultimately get transported to the Golgi. We're going to talk about the Golgi in a second. So you make the polypeptide chain, or the soon-to-be protein, package it into a vesicle, and head it off to the Golgi. The smooth ER, we'll stick with ER since we are, again, talking about endoplasmic reticulum. The smooth ER has some slightly different kind of roles, I imagine. We're going to see a high amount of smooth ER in liver cells, because liver cells are good at detoxifying. And what will happen is the liver is going to break down toxic substances. It's going to help detoxify. It's also going to be found to help lipid production. All of those things will happen in the smooth ER. Here's our Golgi. The Golgi is, again, a great organelle. The Golgi is going to actually receive, so let's put it right there, it's going to receive the vesicle from the rough ER. It's going to then do some modification. It may also help manufacture glycoproteins, glycolipids. Anything that needs to be modified or created will happen there. In that case, a new vesicle will be made, where that vesicle will once again either be headed off to the cell membrane or for export. And that would be, again, hormones, neurotransmitters, anything that is leaving the cell. Now, the mitochondria, we will see a lot more about the mitochondria when we get into cell respiration. But for here, we're talking our big deal, cell respiration right there. It is not part of the endomembrane system, as much as I know you want to do that. It will help generate some ATP that will be used throughout the cell. We know that, not just in the mitochondria, but throughout the cell. It's got its own DNA. It has its own ribosomes. It is a double membrane organelle. It actually can divide When a cell divides, it actually must divide, because if they don't, each time the cell divides, there's going to be way less mitochondria, and ultimately that's going to be a problem. And the cells will use the ATP from the mitochondria throughout the entire cell, not just in the mitochondria. Chloroplasts, same thing, not part of the endomembrane system. In this case, we're going to do photosynthesis. They must and can divide each time the plant cell divides in this case. Again, they have their own DNA, they have their own ribosomes, they are a double membrane organelle, and they can and do produce their own proteins and enzymes. Cell wall is only found in plant cells. That is made up of a non-living material called cellulose, and that is going to be all about cell structure. That is what will keep a plant from bursting if water flows in, and will also give it some integrity if it happens to be low on water. Talking about microtubules, microfilaments, and cytoskeleton. This got literally 30 seconds from us this year, and again, it's going to get 30 seconds now. When I talk about these three, I'm going to lump them in for cell structure or cell shape. That's basically what we're going to deal with them. That's it. They have nothing to do with the cell size. The cell size is going to be determined by the genetics of the cell, and we know, and we're going to find out again soon, that cells cannot get too big. They are very inefficient at large sizes. Phagocytosis, pinocytosis, and exocytosis. Phagocytosis is in essence cellular eating. That's what white blood cells will do. And that's how a white blood cell will engulf something, let's say a pathogen, something that the body doesn't want. At that point, the pathogen is now inside of the white blood cell. The lysosomes will come to town. They will digest that pathogen or whatever the cell does not want, body doesn't want. All is good. If white blood cells are working well, you're working well. Pinocytosis is cellular drinking. It's kind of like non-specific. It's basically picking up already digested material. So it's small things. If you look at phagocytosis, you will notice that that material is large and needs to get digested or broken down with the lysosomes. And anything the cell does not want, it simply gets rid of, and that is what is known as exocytosis. We spent a lot of time talking about cell size and surface area. Oh, shoot, forgot the word area here. Surface area, surface area to volume ratio. And again, the surface area doesn't really matter. The volume doesn't really matter. It's all about the ratio. And the rule of thumb is a small cell versus a large cell. Bigger is not better here. A smaller cell has a higher ratio, and the ratio is good because materials can be either taken in, released, they can be exchanged, whatever word you feel like saying, more quickly when the cell is small. Look at how much time it takes to get to the center of that cell. As important as getting in material is getting out material. So bigger is not better. When you talk about size, if I give you a picture, smaller cells, like this little cube over here, has a very large surface area to volume ratio. It is very good. It is very efficient. Now sometimes, unfortunately, let's say you are a mouse, and we saw this in the lab. If you're a mouse versus an elephant, you're small. And that's great. But if you happen to get stuck in a cold area, you are going to lose heat extremely quickly. So in the case of a mouse, the large surface area to volume ratio, if it gets cold, is not good. But for cells and absorption and all of that within your body, big surface area to volume ratio is good. In essence, small cells. Now here is basic an overview of the cell and all of the goodies. So we've talked about our ribosomes, we have our bound, we have our free, mitochondria, there's the Golgi, again the rough ER has the bound. Cell membrane we talked a lot about, there's that cytoskeleton, is all about shape and structure. Vesicles are things that will transport. Lysosomes are where we have our digestive materials if we need them. The smooth ER right here, that's for our detoxification. Again, found a lot in liver cells. And of course the nucleus, that is where we have our DNA. Now this is an animal cell. A plant cell will look similar. It'll be square in shape. It'll have a large central vacuole. It'll have chloroplasts. It'll have a cell wall. But these are eukaryotic cells. These are all organelles. Oops, spelled it wrong. Organelles. And organelles are all about compartmentalization and efficiency. Now, how does that compare between a prokaryotic and a eukaryotic cell? Prokaryotic cells are simple. They're going to be bacteria. They are going to have a cell membrane. They are going to have ribosomes. They are going to produce proteins. They are going to be able to replicate. But they do not have membrane-bound organelles. They have no nucleus. But they do, however, have DNA. Now, since they don't have a nucleus, the DNA has to live somewhere. And the DNA lives in what is called the nucleoid region. And the nucleoid region, once again, is where the DNA is found. It makes mRNA, but the mRNA does not leave through nuclear pores because it does not have a nucleus. The mRNA that is made will simply go to the ribosomes, and all protein synthesis that needs to occur will. Again, no endomembrane system. They do not have rough ER. They do not have Golgi. They do not even have vesicles. Everything takes place within the cell. Eukaryotic cells are way more complex. Those are the ones we just saw. Membrane-bound organelles, things like nucleus, rough ER, smooth ER, mitochondria, chloroplasts. And basically these organelles allow for the isolation of specific functions, which in essence will allow way greater efficiency within the cell. So although it looks like it has more going on, the efficiency is way better, and these cells can grow larger and do more functions. Now we talked about our friend in the notes, a paramecium. Now paramecium is a little guy like that, primarily lives in fresh water, and it has this kind of star-shaped thing called a central vacuole. Now, if you put a paramecium into fresh water, like we learned about earlier, and there is some actual molar concentration of material inside, water is going to want to flow in. Well, that's not going to be good because this thing does not have a cell wall. So it will swell and burst and the organism will die, and that's bad. So this contractile vacuole, what it does is it fills up with water, then it pumps out the water. More water moves in, vacuole fills up, pumps it out. Keeps everything all set within the single paramecium. If that pump is damaged, however it may be damaged, now the water is going to flow into this paramecium. It has no way of getting that water out, and unfortunately that paramecium will burst. So the contractile vacuole is extremely important. That contractile vacuole will work very hard if the paramecium is in a freshwater scenario. If it happens to find itself in a slightly brackish place, sort of kind of like an intertidal region maybe, then that contractile vacuole does not have to work as hard, because as you change the concentration of the water surrounding the paramecium, less water will move in. So that is pretty amazing how this little guy can live its life. And finally, the last page. We talked a lot recently of the endosymbiotic theory. And basically what that states is primitive cells took up things like mitochondria and chloroplasts, and these mitochondria and chloroplasts, they were able to live on their own prior to being engulfed by eukaryotic cells, and they could do that because of the double membrane, the folded membrane, the DNA, the ribosomes, the enzymes, the proteins. Everything they did was fine. When they got taken up by the primitive eukaryotic cells, all of a sudden, that was a win-win. The mitochondria had a place to live. The cell got the ATP out of it, and it was a win-win. In the world of plants, they took up chloroplasts. Same thing. Great symbiotic relationship. The chloroplasts had a safe place to live. They could divide inside of the cell. And they used the power of the sun for photosynthesis, which, again, helped out the plant cell. And the last thing, which was actually one of the first things we did, the endomembrane system. As a quick refresher, we know we start with the nucleus. We send out the mRNA. That goes to the rough ER. The rough ER is going to send out a vesicle that has the protein in it that needs to be modified. That will be sent to the Golgi, where it will be further modified, put into another vesicle, where that now finalized, modified protein can be put into the cell membrane or exported from the cell. And again, we talked about that a lot. So there is the rundown, and there is the picture. So nucleus, well, you can't see it in blue. Nucleus, the mRNA heads to the rough. There's your vesicle. There's your Golgi. There's your vesicle. And you can either be put into the cell membrane or you can be exported. So we did a lot. We took a big tour of the cell. We did organelles. We did water movement. We did relationships. We did it all. So the good news, it's over. Good luck on your test.